Hello, and welcome to the first set of videos for microbiology, that is Bio 234. So today's lecture is lecture 1, and it has several different topics, and what they all have in common is the rest of the course builds on them, and it's important to know all these things before we move forward. So this is a lot of stuff for one day, but it is important to get this out of the way. Okay, so this is a science class, and if we had a live or face-to-face lab component, it would be more of a science class. As it is, I'm mostly going to be teaching you things that we have discovered about microbiology, not as much the process of how we discover new things. Science is the process of how we discover new things. And a science class should focus on that. We can't really do that because we don't have the typical lab set up here. But that's okay. So I want to start with what science is and what it looks like, and specifically this would be the scientific method. And the scientific method is the set of steps we use to organize scientific activity. I'm sure you've heard this before, you've heard of this, and I bring it up to focus our attention on how it works, but also because a lot of us learn some incorrect things about this. There's a major misconception about the scientific method that high school teachers all over the country have been teaching aggressively for the last 20 years. And it gives people a very skewed idea of what science actually is. So I'm going to get into that here. So the first thing we do in science is we make an observation, and this is where our curiosity takes us into the world, and we think, I want to know more about this thing and how it works or what it is. So this could be natural history, where people go out and measure things, or this could be trying to understand how something works. And in microbiology, that's usually what it is. It's trying to figure out how a cell does something, for example. So we ask a specific question. We say something like, how does E. coli survive in the urinary tract? Or more specifically, how does E. coli find the bladder during a urinary tract infection? How does it know where to go? That's a big question, and the answer to that question will involve a lot of different ideas. But that is the kind of question that we ask. So then we generate a hypothesis, and the hypothesis might be E. coli can sense a certain form of the amino acid serine that is typically found in urine, and it can sense the concentration of that. And if it's swimming in the correct direction, the concentration of that will increase. So that's a hypothesis I could make. That's my understanding of how this works. Notice I didn't make a prediction. I didn't talk about an experiment. I just made a hypothesis. And a hypothesis is just a explanation of a phenomenon. It's just the best explanation I can come up with, and I want to test this explanation. And so I design an experiment. I think about how I could design the experiment, and that would be the next step, designing the experiment. Once I have an experimental design, then I can make a prediction, and I can say, well, if my hypothesis is correct, then in my experiment I will get a certain result. And if my hypothesis is not correct, in this experiment, I will get a different result. The distinction I make here between this and what high school science teachers teach is that we typically in science, we make a hypothesis that is hard to test. And it's not directly tied to any one experiment. We could think of lots of different ways to test a big hypothesis. So we can't combine a hypothesis with a prediction until we've thought of a way to test the hypothesis. So that's I don't know, you may or may not care about that, but you should understand that microbiologists will spend years trying to test one hypothesis because they are big and complicated, and there are a lot of different experiments that might need to be part of it. So then after we do the experiment, we get a result and we can make a conclusion. The things that go into experiments are variables and control groups. I'll talk a little bit more about that in the coming slides, because I want to talk to you a little bit about the history of microbiology and how we came to think about life, and in particular microorganisms, how we came to think about them the way we think about them. So if you talk to a student 200 years ago, they would have a very different idea of things like the common cold, or where oxygen comes from, or really anything that could have to do with microorganisms. So that'll be in the next set of slides. So yeah, get some coffee. We'll be here for a while.